Okay, here we are at chemistry, trying to get you to remember your basic chemistry, or maybe to fill in some gaps that you may not have had or you don't remember. Okay, so I'm going to take you through atomic structure and bonding, chemical equations a little bit, mainly focusing on inorganic and organic chemistry. Okay, basics, just to take you through basic structure. We got over 100 different elements. Let me show you a periodic table of the elements. That's not it. There we are. Okay, this is a really nice little periodic table. It's at ptable for periodic table.com. And there's much more information here than you need, but look right up here. When I pass over these elements, the square over on the top left kind of changes, so you can see the stuff a little bit better. Okay, so I'm over oxygen, and oxygen, you'll notice that there's a number at the top, 8, that's atomic number, and at the bottom there's a 15.9994. That's atomic weight, or commonly called atomic mass. If I go to carbon, you got an atomic number of 6. You got an atomic weight of 12. Let me point out hydrogen's kind of interesting. Its atomic weight and an atomic number are the same, 1. Keep that in mind. Okay, so let me go back over here. All right, so you're looking at helium here. Let me go through the parts of an atom. You got a nucleus, and then you've got electron orbits. The electrons are out here, obviously. In the nucleus, you've got protons and neutrons. So a proton is a positively charged particle. Neutrons have no charge. Electrons have a negative charge. So on my helium molecule here, helium atom, you've got two variations of diagrams of an atom of helium. You see 4 little balls inside of the nucleus. 2 of them are protons, positively charged. 2 of them are neutrons, non-charged. Then you can see an electron orbit right here with 2 little electrons. Electron number is the same as a proton number. So if there are 2 protons here in the nucleus, there are 2 electrons out in the orbit. So you've got 2 positive charges, 2 negative charges, and 2 no charges. So it all equals out. Atoms have no charge on them. And over on the right side, you just see the electron orbit is kind of a electron cloud. Now remember I said hydrogen was kind of interesting. Here's a hydrogen down at the bottom, near to the bottom right. Notice it's got 1 proton in the nucleus. It has no neutron, and it has 1 electron in its orbit. So its weight and its number are the same. Atoms of the same element can exist in different forms. They're called isotopes. So you see here 3 different isotopes of hydrogen. The most common is the one I was just talking about, with an atomic number and an atomic weight of 1. 1 electron, 1 proton, no neutron. But if you look at the center one, you've got deuterium, which has an atomic number of 1, and it's got an atomic weight of 2, because notice that there is a neutron here that's not in the normal, most common hydrogen isotope. That's called deuterium. And then on the right, you can see there are 2 neutrons. So in the nucleus, there are 2 neutrons, 1 proton, so the atomic weight is 3. Its atomic number is 1 because it has 1 proton. And here's an example of oxygen. Your normal run-of-the-mill oxygen has an atomic weight of 16. It's got 8 protons and 8 neutrons. And that makes up almost virtually 100% of all the atoms of oxygen. But you can also have an isotope of oxygen which is 17 atomic weight. It's got an extra neutron. The heavier isotopes tend to be unstable. If they're really unstable, they can generate radioactive rays of different types. They're called radioisotopes. They produce various emissions, alpha, beta, gamma. Just wanted to mention that. Okay, bonds. This is where atoms interact with each other, involved in sharing of electrons of some sort. And when they do that, they form a bond. And they do that in order to make their electron shells stable. I'm not going to get into how many electrons you have in each shell. That's more information than you need. But just remember that atoms bond with other atoms, creating chemical reactions in order to get to stability. So when an atom reacts with or forms a bond with another atom, you have a molecule. You can have a very simple molecule like 2 atoms of oxygen. That's an O2 molecule. Or you can have a couple of hydrogens with an oxygen. That's a water molecule. Or you can get more involved and have C6H12O6, which is a molecule of glucose. And there are 6 carbons, 12 hydrogens, 6 oxygens. Now, compound. Any combination of atoms produces a molecule. But if you have 2 or more elements in your molecule, you've got a compound. So H2O is a compound. CO2 is a compound. C6H12O6 is a compound. O2 is not. It's just a molecule. H2, hydrogen gas. N2, gas. They're all molecules but not compounds. There's a nice chemistry tutorial, and I think this is the University of Arizona. You might take a look at it if you really want a good tutorial. It's got various animations and other things there. Okay, let me take you through the bonds quickly. I mentioned that atoms need stable shells, electron orbits, and they will share electrons in order to get a certain number of electrons in their orbit. So I'm not going to cover all the different kinds of bonds, just major ones. Alright, so if you look over here at this carbon, you've got a carbon atom right here. And notice in the first shell there are 2 electrons. That's a stable first shell. And in the second shell, stability means 8 electrons. Also the third shell is 8 electrons for stability. Well, carbon has an atomic number of 6, meaning that it's got 6 protons, 6 electrons also. And if there are 2 electrons in the first shell, that leaves only 4 electrons in that second shell. And it needs 8 to be stable. So it's going to go looking for 4 more electrons to share in that outer shell. So you see 4 hydrogens right over here. Now think about hydrogen. It's got only 1 electron. It's only got 1 proton too, but anyway, it's got an atomic number of 1. So there's your lone hydrogen. So it needs 2 electrons to be stable in this first shell. So if you take this hydrogen right here and you put it into close proximity with a carbon, one of these outer electron orbit electrons can be shared by the hydrogen atom. The hydrogen is now happy because it's got 2 electrons in the shell. Carbon not so happy because it still has 4 of its own. It's sharing the hydrogen electrons, so it thinks it has 5, but it needs 3 more. Well, let's stick 3 more hydrogens around the area. Okay, so now each one of these hydrogens is sharing one of these electrons that you see in the second shell of carbon. And the carbon is sharing 4 hydrogens, plus it had 4 of its own, so it's got 8 in the second shell. Here's what the carbon-hydrogen combination looks like on the right. This is methane, a molecule of methane gas, CH4. You see a stick representation on the far right. Up here on this other molecule, you see 2 hydrogens covalently bonded. You see oxygen covalently bonded, and so on. All right, so oxygen always covalently bonds. Carbon always covalently bonds. Hydrogen can go either way. These are strong bonds. Examples, you're going to see a lot of them when we cover the organic molecules at the end of the chapter. They're all covalently bonded. Water is kind of interesting, though. Let me go back one step. Look at this oxygen right here. Okay, so oxygen has an atomic number of 8. It has 8 electrons. It's got 2 in the first shell. It's got 6 in the second shell. It needs 2 to be stable. So if we get 2 oxygens together, the second shell, electron shell, has 6 of its own, plus it's sharing 2 that are in the shell of this second oxygen. And the second oxygen is sharing the 2 of this first oxygen. So they're happy together, covalently bonding, pretty much equal sharing of the electrons. The electrons are kind of equidistant from the nucleus of both atoms. So this is about a 50-50 sharing. And because this is a sharing, the O2 molecule that you see here is nonpolar. In other words, there's no charge on the molecule. The left side of the molecule is the same as the right side of the molecule. Okay, now water is different. So now you're looking at a water molecule. So on the diagram right over here, you see oxygen, the big guy in tan, and you have 2 blue hydrogens. So what's going to happen if you look down here at the bottom right? Again, oxygen has 6 electrons in that shell. It needs 2. We throw in 2 hydrogens. Remember, hydrogen only has 1 electron in its shell. It needs 1 to be stable. So it shares, each one of these is sharing 1 of the electrons of oxygen. Oxygen is sharing the electron of each of the hydrogens. Everybody's stable. But something a little bit different happens. And what's different is that the electrons of hydrogen are a little bit closer to the nucleus of oxygen. You don't see that on this picture, but that's what it would look like. So it's not a 50-50, it's more like a 60-40 with the electron of hydrogen being closer to the oxygen. Oxygen is the 60 part, and hydrogen is the 40 part. It's kind of like relationships between men and women in a marriage, where in some marriages you might have each person doing 50% of the work, 50% of the washing dishes, and 50% of the cooking, and 50% of the whatever. But frankly, in most marriages, one person is doing more, mowing lawns, and the other person is doing more cooking. If you had a situation where one person was doing most everything, that's more like this kind of a covalent bond. So because the electrons are closer to the nucleus of the oxygen, and remember that electrons have a negative charge, that means that the side of the molecule on the oxygen side has got a slight negative charge. And over here at the hydrogen end, like between the 2 hydrogens at the right side of the molecule, you've got a slight positive charge because, again, they have 1 proton. Protein's got a positive charge. So if you look at this middle picture on the right here, you see that polarity of the molecule. The little symbol here means sigma, and frankly I can't remember why they chose that. But anyway, you've got a slight negative charge on the oxygen end of the molecule. You've got a slight positive at the other end. This is a polar molecule. Water is polar. Now, this also means that a water molecule is going to be attracted to other polar molecules of opposite charges. You'll see that come up in a minute. So again, if you look at the CO2 molecule right here, the carbon is in black in the middle between the oxygens. This is nonpolar. This is equal sharing of electrons. There's no charge on the end of the molecule either side. Okay, ionic bonding. All right, so let's take this atom of sodium and an atom of chlorine. Sodium has an atomic number of 11. It's got 11 protons. It's got 11 electrons. So we've got a full first electron shell, full second shell, third shell. There's only one lousy electron. It needs 7 more to be stable. So obviously it makes much more sense to give this electron to another atom and just get rid of you don't need that electron shell. So here's chlorine. It's got an atomic number of 17. It's got 2 electrons in its first shell. It's got 8 electrons in its second shell. It's got 7 electrons in its third shell. It needs 1 lousy electron to be stable. So when these guys come together, the electron of sodium ends up in the electron shell of chlorine. In fact, you can see it over here on the right-hand side. They don't really separate. They have kind of a tenuous, weak bond between them. So if you put NaCl into water, the NaCl splits because ionic bonds are very weak. And you end up then, if they split, with ions. So Na, because it's lost one of its electrons, has 1 more proton than electron. It's got 11 protons and only 10 electrons since it gave 1 away. It's got a positive charge to it. And chlorine, since it's got an extra electron, has got a negative charge. So when these guys are bonded together, like in salt molecules in your salt shaker, they're still ionically bonded together weakly. If you looked at one of those atoms, one of those molecules of NaCl, you'd find that it's a polar molecule. One end of the molecule positively charged, one end negatively charged. If you take this NaCl and you add it to water, or add water to it, because NaCl is a polar molecule and water is a polar molecule, they interact. And that's what you see on the picture up here. So here's the NaCl, the odd, I don't know, block with a cone on it. And the cone and the block represent the Na and the Cl coming apart in the water. The water molecules are the green and purple molecules. So opposite charges attract. The negatively charged atom or ion is going to be attracted to the positive end of the water molecule, that is the hydrogen end. The elements that do this, I'll take you through this coming up in a bit. Sodium, potassium, what are called the electrolytes. They're weak bonds. Hydrogen bonds. I've kind of already set this up, I hope. You're looking at water molecules in blue. Remember, these are polar molecules. Now the hydrogen is covalently bonded to the oxygen. We're not talking about that. We're talking about these dotted line bonds between individual water molecules. So the water molecules are kind of attracted to each other. The negative end of an oxygen of a water molecule is attracted to the positive end of a different water molecule next to it. And it forms this kind of cohesive attachment of water molecules. That's the hydrogen bonding. It is not any kind of exchange or sharing or taking or receiving of electrons. It is simply a very weak attraction because of the polarity of the molecule. Remember, it's weak. Examples of molecules that have them. Well, water, DNA, RNA, proteins have got hydrogen bonds between the side groups. So you'll see examples coming up. Oh, here's just a look at examples of different kinds of molecules and what category they fall into. Okay, chemical reactions. Again, it's just chemical bonds being broken or made or rearranged. Here's an example of a very simple chemical reaction. Not going to get into this very much. Different kinds of reactions: anabolic, catabolic, exchange, and oxidation reduction. Anabolic reactions are synthetic reactions. For example, linking amino acids together to form a protein, or sticking two glucose molecules together to make a maltose molecule. That's anabolic. Catabolic reactions are degradation or decomposition reactions. For example, let's say you have a starch molecule and you break it apart into individual glucose molecules. That's catabolic reaction or catabolism. I'm sure you've covered cell respiration. That's a catabolic pathway where glucose is split apart down to carbon dioxide and energy. I'll mention energy later, but just to remind you, anabolic reactions use energy. Catabolic reactions produce energy. Then you got exchange reactions, which I'm not going to get into. And then we've got oxidation reduction reactions, which are giving away, donating, or accepting hydrogen ions and electrons, and you've seen that covered in cell respiration before. Exergonic versus endogonic. I misspelled that. It's supposed to be endergonic. Endergonic means got to have energy input. Exergonic means energy producing. So, here's a reference to energy. Production of ATP, endergonic reactions. The ATP has got to be used to produce muscle contraction. I'm not going to get into that more. Okay. Organic versus inorganic molecules. Let's take something like an O2 molecule. Atmospheric oxygen, O2, is 2 atoms of oxygen. That's inorganic. Water, H2O, inorganic. H2SO4, sulfuric acid, inorganic. So what you should have noticed already is there's no carbon in any of those. So let's look at something like C6H12O6, glucose. It's an organic compound. There's carbon in there. In fact, you've got to have carbon for an organic compound. But it's more than just carbon, because if it was just carbon, CO2 would be an organic compound, but it's not. Got to have hydrogen bonded to carbon. So in that C6H12O6 glucose molecule, many of those hydrogens are attached to the carbon. And that's what makes it an organic compound. You've got to have two things, carbon and carbon-hydrogen bonds. Okay, reminder about water. Universal solvent. Water is a solvent. Anything that goes into solution in water would be a solute, like salt or protein or sugar. When you add solute to a solvent, like water, you get a solution. Water is a universal solvent. More things dissolve in water than any other kind of solvent. I've already talked about hydrogen bonding, so you know that water molecules are attracted to each other, and this causes them to pull together in tight little units, like bunches of kids all holding hands. That's the cohesiveness of a water molecule. It's also the reason why water molecules have surface tension. So, for example, if you take a glass and you fill it with water, and you're careful, up at the top, you can actually keep adding water, so the water actually fills above the top, the rim of the glass. It looks like kind of like a bubble of water if you looked at it exactly from the side. That's surface tension. If you stick your finger in it or a fork, the water will run over the edge. You've broken the cohesiveness of the water molecules. Again, you've broken up the hydrogen bonding. Okay, electrolytes. Features. These are compounds, they're inorganic, that conduct electrical current. When you put an electrolyte in water, because they are made of ionic compounds with ionic bonds, and ionic bonds are weak, the bonds will break, and the components of the electrolyte dissociate into ions, charged atoms. They're called ions, positive or negative. Positively charged ion is called a cation, and negatively charged ion is called an anion. Some electrolytes will dissociate very strongly, some very weakly. I'll show you examples coming up. 3 classes: acids, base, salts. Very quickly, a salt is a combination of a cation and an anion, like Na, sodium, and Cl, chlorine, NaCl. An acid is a proton donor. So when an acid is put into water, it dissociates, and one of the things it dissociates into is hydrogen ion, or also called a hydrogen proton, because the proton is positively charged. It's the only thing in the nucleus of a hydrogen atom. A base accepts hydrogen ion, or it produces hydroxyl ion, which is OH. Alright, pH. The way you determine pH, or the way pH is determined, the numbers that is, it's a logarithmic number. So the formula, you're looking at the concentration of hydrogen ion, moles of hydrogen ion per liter. So neutral pH, 7, if you look at the scale down at the bottom, has 1 times 10 to the minus 7 moles of hydrogen ion per liter. You knock out the negative, you take the exponent of 10, which is 7, and that becomes your pH value. So this is a logarithm. You take that exponent of 7 and that becomes the number. When you go to the left of 7 on this scale, down into the lower numbers, you're in acidity. So if you look at pH 5, look at 10 to the minus 5. That's 1 times 10 to the minus 5 moles of hydrogen ion per liter. Again, forget the minus, you take the 5, and that becomes your pH. 10 to the minus 5 is a larger number than 10 to the minus 7. Okay, now look at 10 to the minus 2. That's an even bigger number. So 1 times 10 to the minus 2 moles of hydrogen ion per liter. That's even got more hydrogen ion than pH of 5, which has more hydrogen ion than pH 7. So the further left you go, the more acidity you have, because the more hydrogen ion concentration. Strong acids down at 0, 1, 2. Weak acids at the 5 and 6 area. So again, if you look up here at the picture, you've got 2 acids, hydrochloric acid, carbonic acid. Hydrochloric acid in water dissociates totally into its 2 ions, hydrogen ion, chlorine ion. Carbonic acid is a weak acid, whereas hydrochloric acid is a strong acid. Carbonic acid, in the pH of like 5 or so, weakly dissociates. So you can see most of it is still attached as carbonic acid, H2CO3, but some of it has dissociated. And of course, bases, the same kind of thing. Less, least hydrogen ion as you get over into the 14 area. So 10 to the minus 14 is the least amount of hydrogen ion on this scale. That's a pH of 14. So strong bases at the end, like 13, 14. Weak bases, pH of 8 or 9, something like that. Buffers neutralize acids or neutralize bases. They keep the pH more towards this central neutral scale. Okay, so now we're at the organic compounds. Before I get into each of the types, just some basics about them generally. They are associated with plants and animals, all covalently bonded. Non-electrolytes. They are not made of ionic bonds. They're covalently bonded. You don't get electrolytes from covalently bonded molecules. The principal bonds are carbon-carbon and carbon-hydrogen. The organic compounds have common characteristics, so let me tell you what they are. They are carbohydrates, lipids, proteins, nucleic acids, 4 of them. And they have common characteristics. They're all polymer molecules. That is, they're all made of monomer units, single units, like a carbohydrate, such as a starch. Starches are made of hundreds and hundreds and hundreds of individual glucose molecules. The monomer is called glucose. You stick a bunch of glucose molecules together, and you get a polymer, which is starch. If you look at the picture on the right-hand side, you've got 2 amino acids, glycine and alanine, forming a dipeptide at the bottom. So in order to make a protein, we just keep adding amino acid by amino acid by amino acid. In order to create a bond between these 2 amino acids, we have to remove water. That's called dehydration synthesis. So look at this hydroxyl on the left-hand amino acid. It's got a white background on it for you to see easily. And the right-hand side amino acid, there's a hydrogen sticking off right here. These 2 are going to split off and form a water molecule. You can see that water molecule down here at the bottom right. It's got a red circle around it. That means that the carbon now needs to share an electron. The nitrogen needs to share an electron. They're going to share each other's electrons. We're going to form a bond between the carbon and nitrogen. There it is right there. That's a covalent bond. So this is called dehydration synthesis. We are synthesizing a larger molecule. We're going from 2 individual amino acids, small molecules, to a larger molecule called a dipeptide. It's called dehydration synthesis because we're removing a water molecule. Now, if we want to go the opposite way, if you have a dipeptide, or let's say you had a disaccharide, like sucrose, and you add water to that, you'll have a split of the bond between the 2 individual sugars that make up a sucrose, and you will, in the end, get the 2 individual sugars separated by adding water. And that would be a fructose and a glucose, because that's what makes up table sugar sucrose. That's called hydrolysis. So if you want to break a larger molecule down into its individual monomer units, you add water. That's hydrolysis. You make all these molecules, all the organic compounds, the same way. So dehydration synthesis works in proteins, works in nucleic acids, works in lipids, and hydrolysis, the same thing. These are all big molecules, macromolecules. Okay, so starting with carbohydrates. Here's a sugar molecule, individual monosaccharide. This is a kind of a chain form that you see right here. You can see it's got a backbone of carbons, 6 of them. Lots of hydrogen sticking off the carbons. Remember, carbon needs to share 4 electrons in its second shell. It's got 4 already. So you see 4 bonds here. So if you look at, I don't know, the second carbon even, right here at the top, it's got a single bond with the first carbon. It's got a single bond with the third carbon. It's got a bond with hydrogen. It's got a bond with oxygen that's forming part of the OH. The hydrogen sticking off of the OH here is actually bonded to oxygen. It's not in any way associated with carbon. But that's a sugar molecule, a monosaccharide right there. If you added all this up, you've got C6H12O6. And if you look at that formula, you'll notice that there's twice as many hydrogens as carbon and oxygen. So you see the formula here, CH2O to the nth, meaning that if we put a 6 here in the N, you would have CH2O parenthesis 6. Well, there's no number on the carbon and there's no number on the oxygen. And when they don't put a number, it just means 1. So if you multiply 6 by the 1 of carbon, you get 6 carbons. You multiply 6 by the 2 of hydrogen, you get 12. You multiply 6 by the 1 of oxygen, you get 6. So we would have, if I stick a 6 right here in the end, we would have a glucose molecule, C6H12O6. You always have twice as many hydrogens as oxygen. Now when you get into the larger carbohydrates, like sucrose, maltose, and so on, you'll notice that it's not exactly 2 to 1 hydrogen over carbon, so don't make that mistake. It's always 2 to 1 hydrogen over oxygen. By the way, this formula of C12, H22, and O11 are all, it's the same formula for all 3 of these disaccharide sugars, sucrose, maltose, lactose. The difference is the orientation of the hydrogens and the hydroxyls on the molecule. Remember I said monomer units, the small units that make up a polymer. Well, for the carbs, it's the monosaccharides. You see a bunch of them down here at the bottom. Some of them are pentose sugars, and some of them are hexose sugars, and you've also got trio sugars and sugars of all sorts, depending on how many carbons are in the chain. So here's your glucose over on the left. And of course, the other important ones would be ribose and deoxyribose on the far right that you'll see in the nucleic acids. Functions of the sugars, of carbohydrates generally: energy, aerobic respiration, cell respiration. But other functions too. Types: monosaccharides, already mentioned. Disaccharides, polysaccharides. Okay, so up here at the top, you see a disaccharide being made. This is sucrose. I put a red box around it. If you look at the left, you see here's a glucose and a fructose. Here's that water molecule being constructed from the hydroxyl of one and the hydrogen of the other. And when that water molecule is removed, which you see, by the way, over here on the far right with the green box around it, that means that this carbon right here that's attached to the oxygen and this oxygen that's part of the hydroxyl need electrons. So now if you look over here at the sucrose, there's that oxygen right there that's forming a bond with that carbon right there. We've got a disaccharide sucrose sugar. This is dehydration synthesis. We can go the opposite way. Add water, split it back down to its 2 sugars. Okay, polysaccharides. Polysaccharides are insoluble. They are large molecules, really large. Glycogen is 5,000 sugar units, 5,000 glucose units. Probably the 2 that are most important to you are starch and glycogen. Starch, of course, is made in plant cells. We eat it. Glycogen is stored in your cells. It's a kind of a cousin of, starch and glycogen are cousins. So if you look down here at the bottom, at the very bottom is starch. You can see units of glucose attached together. Look at the bonding between the molecules here. And then here's glycogen over on the right. It's, again, very close cousins. And cellulose is the guy at the top of this picture. It's got a lot of cross-linking between molecules. We don't have an enzyme to break cellulose down, but we do have the ability to break down glycogen and starch, or amylose. By the way, I showed you, let me go back, two forms of sugar representation. There's the chain form right over here on the top right. There's this ringed compound form that you see in this other picture. But here is another representation of, for example, sucrose. It's called a chair form. So you'll see it different ways. Okay, go into the lipids. Lipids have got carbon, hydrogen, oxygen, and sometimes phosphorus. The lipids can be very different from each other because there are different types. I'm going to start out talking about the neutral fats. They're the most common guys. So just focus on neutral fats. So look at the molecule up here. You see this really long carbon chain. There can be dozens of these, and you'll notice there are all these hydrogens sticking off of them. This is called a fatty acid right here. I'll show you a picture of a neutral fat in just a minute, but let me talk about two different kinds of fatty acids. So if you look at the picture at the right here towards the bottom, you see two different fatty acids. Now they've eliminated a lot, so really all you're looking at is the carbon chains. The carbons are the blue balls. The black things are hydrogens that are covalently bonded on them. Again, if you look back at this large, long molecule, you've got just hydrogens attached to every carbon. This is a saturated fatty acid. I know you've heard that before. On the other hand, if you look at the bottom right molecule, notice that between two of the carbons there's a double bond. These two carbons are sharing an electron between them, and that means, because remember carbon can only share 4 electrons, each of those double bonded carbons has 3 electrons already. It only needs 1. So each of these double bonded carbons is only going to attach to 1 hydrogen, whereas the single guys up here in the saturated fat can attach to 2 hydrogens. Keep in mind, 4. It needs 4. So in other words, an unsaturated fat has less hydrogen. It's easier to break down. And when you have a fatty acid with 2 or more of these double bonds between carbon, you've got a polyunsaturated fat. So you can have a monounsaturated, you can have a polyunsaturated. But unsaturated means fewer hydrogens, easier to degrade. Okay, so that's a neutral fat. Let me show you what it looks like. So look at the neutral fat, this top part of the shot. You see a neutral fat over on the right. Okay, so you see 3 fatty acids. Now, you'll notice that between a couple of the carbons, now remember these hydrogens, H2, there's a hydrogen sticking off the top and a hydrogen sticking off the bottom. This is just a long carbon chain. You'll see ... That means there can be lots of carbons in between. They've just left a lot of it off because it's too big. So you see 3 fatty acids here, and you see a glycerol molecule, this little 3-carbon molecule. It's in kind of tangerine color. Okay, so let's back up and look at how it was constructed. You start out with an individual glycerol molecule. That's the guy in the orangey background. Now, sticking off of the oxygens on the right-hand side of that glycerol, there's a hydroxyl, a second hydroxyl, a third hydroxyl. OH is hydroxyl. Now, we've got 3 individual fatty acids sticking off of this left-hand carbon. You got a hydroxyl, a hydroxyl, a hydroxyl. So what's going to happen is we're going to strip off a hydrogen off the glycerol, stick it with that hydroxyl. We got a water molecule. We're going to do it a second time. We're going to do it a third time. Notice that there are 3 water molecules on the right-hand side. That means we're going to form a covalent bond between this oxygen and that carbon, a second one between this oxygen and that carbon, a third one between this oxygen and that carbon, and there are the newly formed covalent bonds right there. That's your neutral fat or triglyceride. So the individual units of the fats are glycerol and fatty acids. Functions of the neutral fats. God, you eat them all the time. You can use them for energy. Your cells can use them for energy. Bacterial cells use them for energy also. They make up some of the structure of the cell. Depends on whether we're talking bacteria or humans. Lots of different functions, but energy is a major one. Now, there are some other types of lipids. There are phospholipids. Phospholipids have got a phosphate group. So here's a phospholipid at the bottom right. Here's a model in black and red and blue. Here's what it looks like chemically. You got two fatty acid chains. They're in blue background with carbon-carbon-carbon chains. You've got a glycerol molecule with a kind of a lavender background. You've got a phosphate group in purple. So it's called a phospholipid, and it makes up cell membranes of everything. Human cell, bacterial cell. Steroids, just to mention them, and the waxes, like, earwax, beeswax. Also, bacterial cells of certain bacteria can have wax in their walls. So again, you find them mentioned in A&P as well as microbiology. Just to point out steroids, bacteria don't normally make steroids, but of course your human cells do. Steroids are all cholesterol based. So estrogen, progesterone. Cholesterol has this 4-ringed compound structure. The proteins. Carbon, hydrogen, oxygen. Gotta have nitrogen. Gotta have those 4. Some proteins have sulfur in addition. Okay, so the structural unit, the monomer, is called an amino acid. You see it up at the top right. So you got a central carbon here. It's attached to a hydrogen. Over on the right, with a lavender background, you see a carboxylic acid group, or sometimes called a carboxyl group, COOH. Actually, if you were doing this correctly, you should have a double bond between the carbon and the oxygen here. Double bond. And then you have your hydroxyl bonded to the carbon via the oxygen. So, again, this should be a double bond between carbon and oxygen. So again, there would be 4 shared electrons for this carbon. Then the central carbon, let's go over to the left. Over here you got an amino group, NH2 amino. The amino group, if you take the amino and you take the fact that a carboxyl group is acid, you got the name of the molecule, amino acid. Now, back to this central carbon right here. It always has a single hydrogen. At the other side, it's got some side chain. It's called an R group. Now look at these 4 amino acids. Whoops, sorry, my mouse just fell on the floor and came apart. Stick it back together here. If you look at those 4, amino acids, again, a generalized amino acid is on the left-hand side here. If you look at glycine, the side chains are in green, by the way, green background. Remember, there are over 20 different amino acids. They are all exactly the same except for that R group. So look at the backbone of the amino acid. It's identical to this up here at the top except for that R group. So glycine, the smallest, there's the side chain, plain old H. You look at lysine, it's got a very complicated side chain, actually bigger than the main part of the amino acid. And here's a cysteine, which has got a sulfur little side chain. That's the only difference among the amino acids. You stick a couple of amino acids together, you get a dipeptide. It's like sticking 2 monosaccharides together to make a disaccharide. You get 3 or more amino acids together, you got a polypeptide, and then eventually you get up to a protein. Functions of the proteins: structure, cell wall structure, of bacteria anyway. Human cells don't have, animal cells don't have cell walls. But you've got some proteins stuck in cell membranes, as do bacteria. And of course body structure: hair, keratin protein in the skin, collagen, a lot of hormones. Bacteria make various proteins. Enzymes are proteins. The structure of a protein, its three-dimensional shape, is very important. So let me go to that before I get back to denaturation. Proteins are hydrogen-bonded molecules, and they have hydrogen bonds. So if you look at in pink, you see the 4 different structural levels of proteins: primary, secondary, tertiary, and quaternary. Okay, primary. Think about beads on a chain. You know those little round plastic beads that you can stick together. You know, you probably had them as a kid. You could make a necklace or whatever. Think of each little bead as being an amino acid. So we got amino acid 1, 2, 3, and so on. These little links between the beads or the amino acids are covalent bonds. You see a covalent bond right here. It's called a peptide bond between these 2 amino acids. It's called primary structure. It's the sequence of the amino acids, the order of the amino acids. Now, if you, again, think about a necklace with little beads that snap together. All right, and you've got it laying out flat. It's not a necklace. It's not attached yet. It's just lying flat. Okay, now, let's take this beaded necklace and kind of form a spiral. Well, that's secondary structure. It either forms a helical shape that you see over here, or kind of a pleated paper shape. And this is due to hydrogen bonding. Now remember that there's bunches of side chains sticking off of these amino acids. Let me take you back here, remind you about these side chains. And remember hydrogen bonding. Hydrogen, these hydrogens sticking off that are attached to nitrogen, that are attached to oxygen. Remember, even though these are covalent bonds, the electron of hydrogen ends up being closer to the other atom of a different type. And so the hydrogen at the outer end of it, I guess we'll say, has got a slight positive charge. And that means that if you have 2 side chains kind of coming together, and you've got a slight positive charge on one and a slight negative charge on the other, they're going to be attracted to each other. And that's what pulls the protein molecule into this secondary structure. It's the hydrogen bonding attractions between the side chains. Now, in addition to that, you can have folding of the molecule. So again, go back to your chain of beads that we've snapped together, and we've now made a kind of a helical shape of it. And now take it, keep it in its helical shape, but kind of glob it up in your hand and fold it. Now you've got a tertiary structure. That's what you see right here. Many proteins, by the way, each of these levels of structure build so on the previous one. So you have primary structure and secondary structure built on top of that. You can't have a secondary structure without the underlying primary structure. Some proteins stop at secondary structure, like hair is just hair protein is secondary structure. But some proteins end up globbing up. They're called globular proteins. They're folded, and those are tertiary. Enzymes fit that description. Antibody molecules fit that description. They have a tertiary structure. That folded structure of the molecule is extremely important in the function of the molecule. So you can see how the 3-dimensional structure of an enzyme is all important to the way the enzyme works and what kind of substrate it acts on and attaches to and how it catalyzes a reaction. Most proteins don't go beyond a tertiary level, but there are a few proteins that have a quaternary structure. And this is where you've got more than one polypeptide chain. This particular structure that you see here is a hemoglobin molecule, and there are actually 4 different polypeptide chains here. There are 2 lavender and 2 blue, total of 4 chains. That's quaternary structure. Usually there are hydrogen, not hydrogen, there are sulfur bonds holding them together. Again, these are rather rare to have quaternary structure. But when you have proteins that have quaternary and certainly tertiary, if you expose these, or even secondary, if you expose these molecules to extreme environments like acidity, alkalinity, or you heat them, the shape of the molecule falls apart, it changes. And that means that the protein is not functional, and that is called denaturation. It's changing the three-dimensional shape of the protein, which then affects the function of the protein. So, for example, the enzyme may not work anymore in catalyzing a reaction. So that takes us to an enzyme. All right, so you know that an enzyme catalyzes a reaction. I mean, it's a catalyst. Heat is a catalyst. So, for example, if you add sugar granules to water and you put it, you mix it up with a spoon, or like adding sugar to iced tea for that matter, the sugar will go into solution, dissolve, and the catalyst has been physical movement of the molecules. You've been stirring it with a spoon. Or if you heated it a little bit in a microwave, you'd get the sugar going into solution even faster. Well, those are catalysts. That's what I mean by that action. An enzyme is a catalyst too. So what an enzyme does is it reacts with a substrate. Substrate's in orange, I guess we'll call it. The enzyme, which is in green, this green folded molecule, has a particular active site. The active site attaches to the substrate. That's why enzymes are specific for a substrate. They only act on one generally. And that helps the reaction go faster. Enzymes do not make a reaction go that would not normally go. They just speed it up and facilitate the reaction so that you get more of the reaction. Now, because there is a three-dimensional shape of an enzyme, and the enzyme physically fits with the substrate, kind of like a puzzle piece fitting in, there are various things that can block the enzyme and the substrate. They're called inhibitors. Or if you denature the enzyme and the three-dimensional shape of the enzyme falls apart so that its active site is no longer functional, you can see that it's not going to work with the substrate. So here's a look at how enzymes work. It's not enough to just say they catalyze a reaction. You need to know how they work. They facilitate the reaction, which specifically means they decrease the energy of activation. So if you look at the graph up here at the top, you're looking at time on the x-axis. You're looking at the energy required to make the reaction go up the y-axis. The reactants, or the A plus B or whatever the reactants are in your reaction, they have to get over an energy hump. Look at this energy hump, this little mountain of energy that has to be overcome to get the reaction to go. When you're at the top of that energy hump, the reaction will proceed very easily going down. You can see there's an energy drop here. So looking green here at the activation energy, that's the amount of energy needed to go from down here at the bottom, that is far left, to the top of that mountain, the energy hump. That's your energy of activation. Because an enzyme works with a substrate and it facilitates the reaction, the reactants will come in contact with each other more effectively, which means the reaction occurs faster, and that's less energy of activation. That's what an enzyme does. Less energy needed, reaction goes quicker. So some things I've already mentioned. Substrate. The substrate has a particular enzyme. The enzymes have different names. Usually they end in A's, like amylase, sucrase, Not always, though. There could be a coenzyme or a cofactor needed. It could be like a vitamin, or it could be a metal, or an element like magnesium. And variables, you could inhibit the enzyme, as I mentioned. Heat will denature the enzyme, so that's what I mean by variables. pH can change it too. Okay, last group, nucleic acids. Carbon, hydrogen, oxygen, nitrogen, phosphorus. Gotta have all of them. Okay, the monomer units, they're called nucleotides. You see one right here. It's made of three things. You got a phosphate right over here in gold. Phosphate, phosphorus with oxygens. That phosphate group is attached to a sugar. This is a pentose sugar. It's either going to be ribose or deoxyribose. And that sugar is covalently bonded to a nitrogenous base, which you see in kind of a olive green. Those are the three components of a nucleotide: phosphate, sugar, base. All covalent bonding that you see here between all these atoms. We've got two different types of nucleic acids, DNA and RNA. I'll show you differences here between them in a minute. But functions, DNA is of course the genetic code, and RNA is involved in protein synthesis. Differences between DNA and RNA other than function. Well, two different nucleotides needed. You can't use DNA nucleotides to make RNA or vice versa. So a DNA nucleotide, it's got deoxyribose sugar here, and the phosphates are the same. The bases, well, three of them are the same, but there's one base that's different. I'll show you the bases in a minute, but remind you that the nitrogenous bases are uracil, thymine, cytosine, guanine, adenine. There are 5 different nitrogenous bases. DNA has got thymine, adenine, guanine, cytosine. And generally you just use letters C, A, G, T. The RNA nucleotide over here on the right, phosphate's the same. Ribose sugar instead of deoxyribose, the difference is just an oxygen right there. Still a pentose sugar. The base, in this case it's uracil. They've misspelled it. It should be IL instead of EL. RNA has got cytosine, adenine, guanine, but there's no thymine. There's only uracil. Uracil and thymine are close cousins. So there's a major difference between RNA and DNA right there. The other picture you see here, this is the double helix of DNA. The backbone, I'll show you on the next slide, is made of the sugar phosphate. These are all individual nucleotides, so nucleotide here, nucleotide, nucleotide, all the way down, all covalently bonded. The two strands are held together by these complementary bases. You see AT, and there's like dotted lines between them. These are hydrogen bonds. Again, it's an attraction between the polar ends of the molecule, the side chains that have polarity, have a charge, plus being attracted to minus. It holds the two strands together. Remember, there's no actual sharing of electrons at all. So here's a little section of, it could be a DNA, it could be an RNA. Well, in this case it's a DNA because there's thymine right there. Everything in these nucleotides is covalent bond. And between the nucleotides, covalent bonds. So here's nucleotide 1, nucleotide 2, nucleotide 3, nucleotide 4. Between the nucleotides, you've got a covalent bond here between the carbon and the phosphate group. And the second nucleotide here, between this carbon and the phosphate group of the third nucleotide. So everything you see here is covalent bond. And between S and P means sugar and phosphate. So you got 4 nucleotides here. An RNA would look just like this, except again you'd have a ribose sugar and there wouldn't be a thymine right here, there'd be a uracil. Now, in the DNA molecule, well, look down here at the bottom right, you see two base pairs, a tiny little piece of a DNA molecule. So you see the two strands of a DNA molecule. On the left, you got two nucleotides attached to each other, and on the right, you see two nucleotides attached to each other. So look at the top left nucleotide. Here's the phosphate, here's the sugar, there's the base. Right here is the covalent bond between that first nucleotide and the second nucleotide below it, which happens to be a cytosine nucleotide. On the opposite strand across, you've got an anti-parallel strand going the opposite way. It's flipped. Look at the sugars, for example. Look at the phosphate group. It's flipped. Anti-parallel. So there's a phosphate attached to a sugar, attached to a base. This happens to be an adenine base. It fits, well, I don't want to say fit, it hydrogen bonds appropriately with thymine. A and T, adenine, thymine. Hydrogen bonds are the dotted lines. And between cytosine and guanine is hydrogen bonding. So again, if you go up here at the top left, no, top right, very top right, you see the two strands of DNA. Up at the top you see the hydrogen bonding, adenine, thymine, guanine, cytosine. There's your hydrogen bonding in red dotted lines. And of course these are more tightly held together and you get the rest of the double helix here. The two strands are complementary. Again, adenine, thymine, guanine, cytosine. Okay, a cousin of DNA and RNA. It's always covered in the unit on nucleic acids because it's a variation of a nucleotide. So let me remind you. Look at this generic nucleotide. Phosphate group, sugar, it's a pentose sugar, and a nitrogenous base. Okay, let me go back to ATP. Now look at this big ATP molecule here. You got a nitrogenous base, this happens to be adenine. You've got a pentose sugar, this happens to be ribose. And you've got a phosphate group. In fact, you're lucky. You got 3 phosphate groups. This is ATP. So this is a variation of a nucleotide. Adenine, ribose, phosphates, except there are 3 of them. Now, these little funny bonds in between 3 phosphates, it's called a tilde. You've got one on your keyboard on your computer. It means high energy. In fact, you can see the tildes up here between the 3 phosphates on this molecule. So when you break the bond right over here, that delivers a huge amount of energy. You see it right here. That energy can be used then to do cell work, to make a reaction go, for muscle contraction, whatever. When you split one of those phosphates off to get that energy, you have 2 phosphates left. Now you have ADP, adenosine diphosphate, instead of adenosine triphosphate. And then of course you can knock another one off and you've got AMP, adenosine monophosphate. So here's a look at how ATP is involved with things in cells, all cells, bacterial, human, frog, whatever you want. You break these bonds right here. That energy that is released is going to then be used to run synthesis reactions. So the energy from this breakup of ATP right here is then transferred for the requirement of putting a bunch of amino acids together into a protein. Synthesis reactions require energy input. Catabolic reactions produce ATP. So look at the glucose right here at the left. If you follow the reaction down, this is just a very, very simple condensation of aerobic respiration in one step. You get CO2 and water and ATP from the breakdown of glucose, and that ATP, again, is used to run synthesis reactions. And that is chemistry in a nutshell. About 40 minutes, I guess. Thanks for paying attention.